Good evening, ladies and gentlemen. My name is Leonardo Fritz from the University of Rome, Tor Vergata, and I'll be presenting our study on thermal resistance to prolonged marine heat waves in four key bivalve species. Marine heat waves are extreme climate events of abnormally high temperatures, generally defined as a period of 5 or more days during which seawater temperature exceeds the 90th percentile of the long-term average for a given location. Climate change is exacerbating the frequency and intensity of marine heat waves with significant effects on marine ecosystem functioning and dynamics. As one of the most relevant abiotic factors, temperature regime strongly affects marine communities in terms of structure and functionality. Benthic invertebrates, such as bivalves, are particularly vulnerable to extreme temperatures as they are sessile and cannot move, thus representing a strong model for the predictive analysis of benthic communities' responsiveness to marine heat waves. Hard substrate bivalves such as oysters and mussels are keystone species that provide numerous ecosystem services. First of all, they act as ecosystem engineer species, as they significantly modify the environment they inhabit. In particular, they act as habitat-forming species since they tend to colonize vast surfaces in mass, and consequently enhance the substrate's tridimensional complexity. As a result, these monospecific associations actually host and sustain a wide range of other species, effectively acting as biodiversity hotspots. Oyster reefs and mussel beds are among the most common and well-known examples of this kind of habitat. For similar reasons, these bioconstructions help mitigate coastal erosion by greatly dissipating wave impact energy. And furthermore, as filter feeders, hard substrate bivalves greatly contribute to benthic pelagic coupling, establishing a trophic link between benthic and pelagic food webs. They also act as carbon sinks, both by building their shells with calcium carbonate and by forming carbon-rich biodeposits through the production of faeces and pseudofeces. Bivalves also play a fundamental role in nutrient cycling as they convert large planktonic biomasses into detritus. This process helps eutrophication control and bioremediation and holds a general utility in enhancing water quality. Many bivalve species also have a great economic value, being exploited in the wild, as well as being highly sustainable aquaculture products, for example from mussel and oyster farming. As a matter of fact, in the European Union, mussels and oysters alone account for 45% of the total biomass produced by aquaculture. This kind of aquaculture is also considered restorative as it both uses and enhances seed resources from wild stocks. Despite their ecological and economic sustainability, few studies have examined their responsiveness to prolonged and continuous exposure to heat waves. In this study, we examined the effects of prolonged marine heat waves on four key species of oysters and mussels, widely distributed in the Atlantic and Mediterranean area. The oysters we chose as target species are the European flat oyster, Ostrea edulis, and the Pacific oyster, Magellana gigas. The European flat oyster, Ostrea edulis, is native to the eastern Atlantic coast from the British Isles to Morocco and the Mediterranean basin. It can be found in estuarine and coastal waters where individuals stick to hard surfaces by releasing cement and can form reefs through vast assemblages. It's been largely harvested and farmed throughout European history up until the last century, but populations have faced a drastic decline in the last decades. Magellana gigas, the Pacific oyster, is a species native to the Pacific Ocean, but it has been introduced for aquaculture purposes in many other areas, including the Atlantic and Mediterranean, often replacing the native Ostrea edulis. The mussels we chose as target species are the Mediterranean mussel, Mytilus galloprovincialis, and the bearded horse mussel, Modiolus barbatus. Mytilus galloprovincialis is a typical Mediterranean species. It's common in the intertidal zone, where it typically colonizes hard surfaces thanks to its byssus, thus forming vast mussel beds. It's largely employed in aquaculture throughout its whole range, and it's been introduced in many other areas of the world. Modiolus barbatus is a Mediterranean species as well, and it also attaches to hard surfaces through its byssus, living generally a bit deeper than Mytilus galloprovincialis. All selected species depth ranges typically include the first 100 meters from sea surface, with oysters usually not exceeding 80 meters depth and mussels sporadically reaching 200 meters depth. For this study, specimens were collected from stable facilities and were transported to the Laboratory of Experimental Ecology and Aquaculture in the lowest possible stress conditions. After a period of acclimatization, individuals were cleared from debris and amphibians and were later positioned in the tanks of a recirculating aquaculture system. Here, maintenance took place as the animals were fed daily and monitored. The experiment began after a 48-hour starvation phase. For the experiment, standard groups of each species were exposed to different temperature treatments, 25, 27, and 29 degrees Celsius, representing respectively a control group, a moderate heatwave scenario, and a severe heatwave scenario. Each treatment was carried out in a single thermostatic chamber and administered to 33 individuals of each species. Every single specimen was positioned in its own mesocosm in order to avoid cascade effect related group mortality. Water changes occurred daily, and temperatures and salinity were monitored and remained constant. Entering water was preheated to target temperature before executing water changes in order to avoid temperature fluctuations. Exposure to treatments was continuous and lasted 20 days, over the course of which mortality was recorded every 12 hours. At the end of the experiment, mortality curves were plotted with mortality percentage as a function of time of exposure, and ET50, meaning exposure time causing 50% mortality, was determined. For Ostreadulis, the European flat oyster, at 25 degrees Celsius, ET50 was 7 days, while at 27 degrees and 29 degrees, ET50 was respectively 2.5 days and 2 days. The 25 degrees group never reached 100% mortality, while the 27 and 29 degrees groups followed a very similar trend in the first 2 days, after which mortality in the 27 degrees group started following a less steep curve and stabilized just before 90%, while mortality in the 29 degrees group grew faster and reached 100% at day 14.5. Magallana gigas never reached 50% mortality at 25 degrees Celsius and stopped at around 40%. The 27 degrees group had an ET50 of 6.20 to 25 days, and mortality rate reached a 75% maximum. For the 29 degrees group, ET50 was 3.7 days, and the mortality curve was steeper but reached 100% only at day 20. Under all tested scenarios, Magallana gigas, the Pacific oyster, appeared to be the most resistant oyster species. For Mytilus galaprovincialis, the Mediterranean mussel, ET50 at 25 degrees was never reached, and mortality was overall very low, reaching 6% at day 20. The 27 degrees group never reached 50% mortality, stopping at around 30%. The 29 degrees group followed a steeper curve, and its ET50 was 7.1 days. However, mortality reached only around 80% after 20 days of exposure at 29 degrees. Modiolus barbatus, the bearded horse mussel, followed a similar trend at 25 and 27 degrees up until day 10, following which the 25 degrees group mortality curve stabilized at 25%, while the 27 degrees group's mortality curve kept growing until about 40%. In both cases, ET50 was never reached, unlike in the 29 degrees group, where mortality reached 50% at day 5.4 and then stopped at around 90%. Under all tested scenarios, Mytilus galaprovincialis, the Mediterranean mussel, appeared to be the most resistant mussel species. Overall, Mytilus galaprovincialis resulted as the most resistant species, followed by Modiolus barbatus and Magallana gigas respectively, and Ostreadulis was the most sensitive species. In general, target species appeared highly vulnerable to marine heatwave events of 27 and 29 degrees. In a changing climate, Mediterranean water surfaces easily surpass 25 degrees Celsius for periods of several days during the summer. posing a threat to keystone species that hold high ecological and economic value. Earth climate observation programs, such as the EU Copernicus, gather vast amounts of global data from satellite, airborne, and seaborne measurement systems and actively monitor, map, and forecast marine heatwave events. Coupled with these information systems, this study's results provide essential data that can be used to evaluate the extension and magnitude of effects of marine heatwave events on bivalve and benthic communities in specific areas and timescales. In the aquaculture sector, the study's results can be used to determine viable low marine heatwave likeliness zones and to establish climate-smart bivalve productions by adapting location choice to the target species performance and limits in different areas. Ladies and gentlemen, I thank you for your attention and I wish you an insightful and productive meeting.